Good evening to you all, and welcome back to SCT 213, Intermediary Metabolism 1. I think we finished with um, our revision on proteins, looking at the structure of amino acids and the different levels of protein organization, looking at the primary structure, secondary structure, tertiary structure, and the quaternary structure of proteins. And in the last lecture, we looked at the function of proteins as enzymes. So now we can go into the metabolism of proteins. Today we're taking catabolism of proteins, which is the breakdown of proteins. But specifically, what we are going to look at is a build-up of a knowledge that you already have, even from primary school, that you have known that proteins break down into amino acids. So that one is clear. Now that you know the structure of proteins to be having amino acids that are having peptide bonds or amide linkages between them, it is clear that for you to get amino acids, it involves the breakdown of the peptide bonds between the amino acids. Further, what we are going to look at today, we can call it catabolism of amino acids, which means now we are coming to look at the breakdown of the amino acids. Because you've already known, you've already done breakdown of proteins into amino acids, which is simple. Just an enzyme adds on peptide bond and breaks peptide bond between amino acids, and then you have the single amino acids. But after that, what happens? What happens to the amino acids? That's what we are going to look at. So we are looking at metabolism of the amino acids. We will start with a poem entitled "The Amino Acids Speak." And it goes like this. We transaminate and deaminate to liberate ammonia that is detoxified in the liver to end product, urea. Greatly important to the body are our nitrogen products. Carbon skeleton is for glucose, fat, or fuel. This poem actually summarizes the whole process of the breakdown of amino acids. So we're going to look at two major processes by which amino acids are broken down: transamination and deamination. And the product that is liberated from those two processes is ammonia. And that ammonia goes into the production of urea in the liver, which later is excreted out of the body in urine. And you also know that amino acids have an amine group in them. That amine group has nitrogen there. So when you have the amine group being removed, that nitrogen can go into the production of other nitrogen products like the nitrogenous bases that are found in nucleotides, which form nucleic acids, or even the nucleotides that you have seen to be coenzymes, like the nicotinamide dinucleotides flavin adenine dinucleotides. Those are some coenzymes which you saw, and even some others like folic acid also contains some nitrogen. So many of these vitamin Bs that act as coenzymes, you will see they have some nitrogen content in them. Now you remember that the structure of all monosaccharides is such that you have an alpha carbon on which you have a carboxylic acid group and an amine group, then a side chain, then a hydrogen. Now, if you look at that structure, then you realize that you have a carbon skeleton on which you are having an amine group as well. So if the amine group is removed, you are left with a carbon skeleton. And that carbon skeleton can go into the synthesis of glucose in a process which we refer to as gluconeogenesis. Or even fats. That is, if you take a look at the structure of fatty acids, you see the structure of fatty acids is just a long chain hydrocarbon with a carboxylic acid group. That's a fatty acid. And that fatty acid, the glucose or carbohydrates which are being synthesized, can be broken down to generate energy. That is what we refer to as the fuel. So that is the summary of the breakdown of the amino acids. Now we can look at this further to say that if you take any amino acid, that amino acid undergoes, say, the process of transamination to form a keto acid. Now, in that process, the amine group, you know, the word transamination is referring to the fact that there is a transfer of the amine group. So there's a transfer of the amine group onto alpha ketoglutarate. We've actually seen this reaction when we were looking at the classification of the enzymes. We saw the transfer of amine group from an amino acid onto alpha ketoglutarate, and that formed glutamate or glutamic acid. And then from glutamic acid, there will be the removal of that amine group, which goes into the synthesis of urea. That process is what is referred to as deamination. We will actually see that reaction I'm referring to. We'll see it again here so that you have some sort of revision of it. You remember that, yes, we saw that reaction where there was transfer of an amine group from amino acid onto alpha ketoglutarate to give you glutamic acid. When we were looking at transferases, you know, we did classification of enzymes, oxidoreductases, transferases, where that was the second class that we looked at before we went on to look at hydrolysis, lyases, isomerases, and ligases. So when we're looking at transferases, so if you go back and look at the last slides, the presentations that we studied the last time, you look at transferases and you will see the reaction that is there as an example. So that is the reaction which is involved in the process of transamination. Now, if we, theoretically, or even it is practical, at any moment in time, if we refer to the fact that at any moment in time, we have some amount of, free amino acids in the body. That amount of free amino acids in the body at any point in time we refer to as amino acid pool. It isn't that like there's a compartment in the body where you have amino acids over there. No. I mean, these amino acids can be found at any position, anywhere in the body at any time. But we are just referring to, collectively, all the amino acids that will be in the body at any point in time, referring to it as amino acid pool. Now that's estimated to be about 100 grams in an adult. Now, where will these amino acids come from? The amino acids will come from food, that is dietary protein. When we eat, the proteins that we take in get broken down into amino acids, which is clear. But we also studied that there are some proteins, there are some amino acids. There are some amino acids that we refer to as essential amino acids. And some of them, we refer to them as non-essential amino acids. The essential amino acids are the ones which we get from diet, from food, because the body cannot synthesize these amino acids. The body cannot produce it on itself. So we have to get it from food. Now, the non-essential amino acids, they can be synthesized in the body. So that's another source of the amino acids going into the amino acid pool. It's either from food, which is a dietary protein, or it is coming from the synthesis of non-essential amino acids. Now, in that cycle, the amino acids that we have in the body, first of all, can go into protein synthesis. I think even in first year, you did something on protein synthesis, where you were looking at the process of replication and then transcription of the DNA onto a messenger RNA, and then the messenger RNA moves to the cytoplasm. Then you have the process of protein synthesis. So you ask yourself, where will the amino acids come from in that process of protein synthesis, where you are having the amino acids joining to give you a protein? So those amino acids are coming from this amino acid pool, either from diet or from the synthesis of the non-essential amino acids. So amino acid can go into synthesis, and that protein, the protein which is synthesized by the body, can also undergo breakdown, catabolism. So there is that cycle where you have amino acids forming protein, and the proteins breaking down to give you amino acids. So the cycle goes on. You have that cycle. Then you also can have these amino acids undergoing, a breakdown, what we'll actually be looking at more into detail, where you have the process of, say, deamination, and that is going to form urea, which will mostly be excreted out of the body in urine. And so you'll be left with just the carbon chain. That carbon chain can then go into the synthesis of carbohydrates or fats, like fatty acids. So it's a carbon chain. So these carbons can go into synthesis. They can come together. That is anabolism. Now, you can also have synthesis of the nitrogen containing compounds, which we're referring to. Like the purines and pyrimidines, those are the nitrogenous bases that you find in the nucleotides that form nucleic acids, RNA, DNA. You can also have these amino acids going into the synthesis of, say, what you refer to as porphyrins. Porphyrins are, you have these 4 rings that are bound together. That's what actually forms the cyclic structure of heme, what you find in hemoglobin. So that will be the prosthetic part of hemoglobin. Other, maybe a byproduct like creatine, which will also be excreted in urine. Or you can even have the synthesis of phospholipids, which forms the membrane of cells, so cell membranes. So the protein can go through different pathways, or just, giving out energy. Of course, that will be coming from the carbohydrates too, because it is the carbohydrates that will then undergo, processes of energy generation, what you have known to be glycolysis, and then the TCA cycle. So this actually gives you like where the source, you have a source of the amino acids, and then you have the use, the utilization. That is the end product that the body makes use of. And then in the middle, you are having that cycle of the amino acids going into the synthesis and breakdown, and part of it, getting lost in the breakdown, where you have the release of ammonia going into the synthesis of urea, and then that is excreted through urine. Now, we can take a look at some general aspects of amino acid metabolism, building on that. So we have just seen that amino acids are gotten from, say, dietary protein. And we also get the amino acids from the proteins that are, synthesized. So that is to say that you have, that process of protein synthesis that I was referring to, where you have the messenger RNA coming from the cytoplasm, what you did in first year, and then a transfer RNA is bringing the amino acids one after the other to form proteins. Those proteins are synthesized in the body. So you can get amino acids from the dietary proteins, what you eat, and the proteins that are synthesized in the body. And those amino acids can be recycled. So they go back into the synthesis of proteins again. Or you will have them going into the synthesis of nitrogenous compounds, or what you also refer to as azo compounds, which we talked about, like the nitrogenous bases, porphyrins, some coenzymes like the nicotinamide and flavin adenine dinucleotides. These amino acids will also undergo the process of transamination, where there is a transfer of the amine group onto alpha ketoglutarate to give you glutamate or glutamic acid. And when that happens, it means you are going to get keto acids from the amino acids. Whilst from the glutamic acid, the process of deamination occurs where there will be a removal of the amine group. And that ammonia then goes into the synthesis of urea, which is excreted out of the body. But these keto acids can go into energy generation. Some of it can go into the synthesis of carbohydrates, glucose. Some of it will go into the synthesis of fats, which is particularly fatty acids. And then you have the rest going into the synthesis of non-essential amino acids that the body can synthesize. Now, going back to the reaction we saw when we were referring to transferases. What did we see? So we saw an amino acid. So we are looking at an amino acid. We call it alpha amino acid 1. And then we have an alpha keto acid. Now pay attention to the side chains, R1. And then there is R2 for the alpha keto acid. And see what is happening. You see that on that alpha carbon, where you have the amine group, for R1, where the side chain is R1, you now see that there is a transfer of this ketone group from the alpha keto acid onto that, in place of the amine group in the alpha amino acid app. You now have an alpha keto acid. And you also have the transfer of the amine group onto the alpha carbon of that alpha keto acid, so that you now have an alpha amino acid. Like, there are two things happening. There is an exchange between the amine group and the ketone group. So there's that exchange. So intentionally, we're showing this with different colors, red and blue, so that you can see the crisscross. There is that crisscross in order that you see the alpha amino acid is forming an alpha keto acid, whilst the alpha keto acid is forming an alpha amino acid because of the transfer of the two groups, the amine group and the ketone group. There's the exchange of the two groups between them. We've seen it before, and it's the same thing which we are looking at. And we actually refer to the enzyme as transaminase. Right, so it is the same reaction. So if you look at it further, you see that is an amino acid with the amine group. So amino acid 1 with the amine group. This amine group, when it is given out, when it's transferred, on that carbon, you now see there is a ketone group. So from amino acid 1, you have the formation of keto acid 1. Now you see, in that process, you now will have a keto acid 2 and amino acid 2, because where is this amino acid going? This amino acid was being transferred onto the keto acid too, so that the keto acid now receives the amine group and becomes amino acid 2. And the enzyme that is involved is transaminase. And what's the coenzyme? PLP there refers to pyridoxal phosphate. And I think we saw one of the coenzymes being pyridoxamine. And I think pyridoxamine also was one of the vitamin Bs. I think vitamin B6 also. I think so, vitamin B6. And I even explained to you that that vitamin is in its inactive form. And it is just the phosphorylation of its addition of a phosphate group to it that makes it active. So from pyridoxamine, you have pyridoxal phosphate as the active form of that vitamin, which is the coenzyme to transaminase. So that is the process of transamination. We can take a specific amino acid. Because so far we just looked at R1, R2, just referring to a general structure of amino acids. But if we take a specific amino acid like alanine as an example, then we will see clearly what's happening. So that's alanine. Remember, alanine has a side chain, CH3. So on the alpha carbon, you have your carboxylic acid group, the amine group, and then the side chain, CH3. Now we see the enzyme, glutamate pyruvate transaminase. From the substrate, alanine, amine group is transferred. And where is the amine group transferred to? The amine group is transferred onto the coenzyme. So you see, the coenzyme and the enzyme are acting together. Remember, the coenzyme is the prosthetic part of the enzyme. And on that coenzyme, you see, there is that ketone group, the C double bond, not a ketone group, as we call, let's say that's like an aldehyde group. And so let's focus on that C double bond O. On that C double bond O there, in place of it, you have the NH3, having that transfer onto that. So from pyridoxal phosphate, you now have pyridoxamine phosphate. The addition of the amine group to pyridoxal phosphate now makes it pyridoxamine phosphate. And once that amine group is transferred, you see there is the ketone group. I mean, the C double bond O from that pyridoxal phosphate onto pyruvate, in place of that amine group in alanine, you now have pyruvate. And you have seen pyruvate, like pyruvate will go into the tricarboxylic acid cycle. So that cycle will give you energy. So you see, the tricarboxylic acid cycle, you saw it in the metabolism of carbohydrates. Now you are looking at metabolism of proteins, and you are seeing the synthesis of pyruvate. So you see the interconnection between metabolism of proteins and metabolism of carbohydrates. In fact, you will see that all of these metabolism of the biomolecules that we are looking at, the proteins, lipids, the fatty acids, you see, they will all be connecting onto the tricarboxylic acid cycle. We saw already transfer with alpha ketoglutarate, which is what we are seeing here. Again, clearly, alpha ketoglutarate is an intermediate in the TCA cycle. Now we see, where is the amine group going from pyridoxamine phosphate? The amine group is then transferred onto the alpha ketoglutarate. And from there, the alpha ketoglutarate, upon receiving that amine group, is then now forming glutamic acid or glutamate. So in place of that C double bond O on alpha ketoglutarate, you now have the amine group. It is the same reaction we are looking at, the exchange, except that this time around, we are looking at it with the participation of the coenzyme pyridoxal phosphate, which means pyridoxal phosphate is that part of the enzyme which is receiving the amine group. So first it receives the amine group before it transfers it onto alpha ketoglutarate to form glutamate, glutamic acid. Now, in other words, you can look at it like this, that it means that glutamic acid is playing a very important role in the metabolism of amino acids or proteins, such that it is glutamic acid that's like a gateway of deamination, removal of amine groups. Because all the time you are seeing that the amine group from an amino acid is transferred onto a carbohydrate, which is also playing an important role there. So together with glutamic acid, they are responsible for the removal of the amine group from the amino acids, the process of transamination and deamination. You see that first amine group goes onto alpha ketoglutarate, then that alpha ketoglutarate, after receives the amine group, becomes glutamic acid. Then glutamic acid now undergoes the process of deamination. The amine group is removed. And you are also seeing the important function of the coenzyme vitamin B6, pyridoxal phosphate. And pyridoxal phosphate is that coenzyme that will receive the amine group from an amino acid before transferring it onto alpha ketoglutarate. So these are very important processes, structures, step by step, that you should pay attention to. You write the reaction that an amino acid, like you see, if you were just writing a simple reaction, you would just write alanine, and you draw an arrow, pyruvate. Maybe if you want to show more, you write in the middle, glutamate, pyruvate, transaminase, or maybe you just write transaminase, just to show that the amine group is transferred from alanine onto another molecule, but the product is pyruvate. That's a very simple, simplified reaction that you write. But what happens in the middle, step by step, before that occurs, is what we are looking at. Step by step, what happens. Now, we can go on and look at what that actually means. I mean, that, we have different we have different mechanisms of action of enzymes. And this particular mechanism that we just looked at, this particular mechanism that we just saw, we refer to it as ping pong catalysis. So you see, it's like ping pong is what? Like table tennis. You play and then the other person plays back to you. Ping pong catalyst. So it's like a table tennis where now you see this is, say, the enzyme. This is the enzyme. Remember, the enzyme is made up of the apoenzyme and the coenzyme, the prosthetic part, which come together to form holoenzyme. That is the active enzyme. So even though we refer particularly to the coenzyme, pyridoxal phosphate, it is part of the enzyme. So if this is the substrate, which is, having that double bond O, and then the enzyme, that is having that NH2. We could actually flip it the other way so that we see that we have the ketone group transferred onto the enzyme, like that's the coenzyme. And then the amine group comes onto the substrate B. There are two substrates. You are having the alpha ketoglutarate. So in this case, this will be the alpha ketoglutarate A, which is having the ketone group. And then it is the enzyme, the coenzyme, that receives that amine group and transfers it. So then you have the amine group transferred to form the how do you call it, the second amino acid, which you saw, amino acid 2. And the enzyme rather is now receiving the ketone group. And you see that sort of amine group, what we saw, it was like you have an amino acid transferring the amine group onto the coenzyme or the enzyme. That's like playing the table tennis to it. And then it also places it onto the alpha ketoglutarate. Then that alpha ketoglutarate becomes glutamic acid before there is the process of deamination. So because of that kind of transfer of the amine group from one molecule to the other, it is referred to as ping pong catalysis. That's like the enzyme is flipping between two states. At one point, the enzyme, so we can go back and look at it. At one point, you see the enzyme looking particularly at pyridoxal phosphate. At one time, it is having a C double bond O. At another time, it is having the NH2 on it and becomes pyridoxamine phosphate. So it's undergoing that kind of cycle. One time, amine group on it. Another time, amine group gone. It has a ketone group on it. Like that, the C double bond O group on it. So on and on. It's like playing the table tennis to and from, to and fro, to and fro, to and fro. That's why it's being referred to as a ping pong catalysis. All right. Now we can move to the next process, which is deamination. Deamination. Actually, if you look at the process of transamination and deamination, they're actually occurring, simultaneously at the same time. And so because of that, even though we talk of it as if they are two separate processes. Sometimes you see that the whole process of the breakdown, the catabolism of amino acids is referred to as trans-deamination. Like it's not separated like transamination and then deamination. They just refer to it as trans-deamination. So it's looked at as a whole because you can see clearly that the process is occurring at the same time. So you can see the interconnection. First, the amine group is transferred. And quickly, that amine group is removed from glutamic acid or glutamate. So there is that simultaneous occurrence of these two processes of transamination and deamination. But of course, there's difference between the two of them. Even in the name, you can see it that transamination is referring to transfer of the amine group. So transfer of the amine group from an amino acid onto a keto acid, particularly alpha ketoglutarate. And deamination is referring to removal of the amine group from glutamic acid. So these are two different processes. Trans means transfer, D means removal. So they are two different processes, except that both of them are occurring simultaneously at the same time. And so it is sometimes referred to as trans-deamination. And you come to see that such simultaneous process, results into, keto acids in the end, like you are having carbon chains. Because once the amine group is removed, the amine group is no longer there. What is left is going to be a carbon chain with some ketone group on it. And those are going to be keto acids. What we saw earlier to be going into the synthesis of carbohydrates and fatty acids, which will then go into energy generation. Now we can take a look at two forms of deamination. The first is oxidative deamination, and the second is non-oxidative deamination. So clearly, if you look at the two names, it just tells you oxidative deamination is going to involve some oxidation reduction reactions, redox reactions, but non-oxidative deamination will not have such a reaction. It just be removal of the amine group without any oxidation and reduction reactions. So let's look at first oxidative deamination. And we're looking at glutamate. Remember, it is glutamate that received, it was alpha ketoglutarate that received the amine group and became glutamate. The product that was formed in that process was glutamate. So that's what you see here, that at the ends, at the left side, you see L-glutamate. At the right side, you see alpha ketoglutarate. And take note that the arrows are reversible. So these reactions are reversible because you see the amine group can be given out from glutamate, and then you form alpha ketoglutarate. Alpha ketoglutarate can receive amine group, and then the product that is formed is L-glutamate. We saw that already with that cycle. Here we are seeing that in that process of the removal of the amine group, and you see the amine group is bold in L-glutamate. In that process, what do we see? We see there is some nicotinamide adenine dinucleotide acting in that process. And the enzyme which is acting there is glutamate dehydrogenase. GDH, glutamate dehydrogenase. And remember, this is deamination, removal. When we were looking at the transamination, the enzyme was what? Transaminase. Here it is glutamate dehydrogenase. So that is an oxidoreductase. And we see these coenzymes which are involved in oxidation reduction reactions. And clearly you can see NADPH plus is getting, there's some hydrogen or proton transfer onto it from that amine group. So you now have an NADPH and then an H plus. You have some transition states, alpha aminoglutarate. So in place of that amine group, what do you see? You now have a C double bond NH. And the reaction continues further with the removal of that NH group there. And in place of it, you are having a C double bond O, and you have the amino group that is removed in that process. Of course, along the line, water comes in that reaction. Then you now have your alpha ketoglutarate. So glutamate, after losing or after the amine group is removed from it, will form the alpha ketoglutarate back. So the reaction continues. Amine group is removed. Alpha ketoglutarate is formed. Now you may be wondering, so why all of this first transamination, later why is the amine group being removed and all of that? You know, you remember that when you were studying things like carbohydrates, you refer to storage form of carbohydrates. In plants, it was starch, and in animals, let's say in us, carbohydrate is stored as glycogen in the liver. How about fats and oils? So yes, if you take plants, you have oils that are stored in the cell membranes. I mean, there's plenty of oil in like nuts, for instance. And that's where we do extraction of oils from a lot of times. And well, there are some plants with their leaves having some oils in them also that are also extracted, or seeds. They have the seeds that oils are extracted from. And in animals, where do we have the storage of fats? Like in us, it's in the adipose tissue. And it's stored as what? Triacylglycerols. You have glycerol and you have fatty acids, 3 of them bound onto it. That's the storage form. Because if you have 3 fatty acids that are bonded together onto one backbone as a triacylglycerol, that's a form of packing of the fatty acids. So fatty acids can be stored as triacylglycerols in both plants and animals. The same way as you can have carbohydrates is being stored in plants as a starch, either in the seeds, like what you find in the grains, or in the roots. So you have like root tubers, or we have some stem tubers, like potatoes, yam, for instance. Now let me ask, what's the storage form of proteins? And you think about it. There is no storage form of proteins, because proteins if they are in the body above the normal levels, they rather become toxic. Why? Because of these amine groups. That is why the amine group is removed. Amine group, when it's removed, is what? Is either coming out as ammonia gas, like bad odor, or it just goes into the synthesis of urea. That's going to be excreted as urine. So you are left with just a carbon chain. It's the carbon chain that will go back into the synthesis of carbohydrates. And those carbohydrates then will go again back to, say, the adipose tissue, or they can go into the synthesis of fatty acids. That one too will go to the adipose tissue, or those carbohydrates stored in the liver as glycogen. So yes, excessive intake of carbohydrates, a lot of it comes out as waste. The rest of it is just going to increase storage in the liver and the fat storage in the adipose tissue. That's what it will just be adding to that. So the amine group is toxic and has to be removed from the body. Right. Now, oxidative deamination, occurs in the liver and in the kidneys. I just talk much about, the storage of carbohydrates and lipids in the liver, and you have these amine groups that are removed in the form of ammonia. And so if you have oxidative deamination of these amino acids occurring in the liver, in the end, those amine groups go into the synthesis of urea, which then goes into the synthesis of urine in the kidneys, and then gets excreted out of the body. And we have seen already what I just mentioned was the purpose of all of these. You are having urea synthesis. So urea will be excreted out of the body, and there will be the production of keto acids. And those keto acids will then go into the synthesis of fatty acids and carbohydrates. We have seen the role of glutamate dehydrogenase in that process. And I mentioned, we've seen in the classification of enzymes that dehydrogenases are oxidoreductases. So dehydrogenase is involved in that process of deamination, the reaction which we saw just before this slide. That dehydrogenase, that enzyme, is regulated by activators such as adenosine diphosphate, guanosine diphosphate, and inhibited by the triphosphates and some steroid and thyroid hormones. Now, particularly about ADP, GDP, you see, you are having glutamate dehydrogenase, which is, removing the amine group from amino acids, and you are left with a carbon chain, which is the keto acids that are formed. And those keto acids will go into the synthesis of carbohydrates and lipids, which will go into energy generation, which is why high amounts of adenosine diphosphate and guanosine diphosphate will be activators. Because what it means is that, you see, the cellular energy is what? It's in the form of ATP, the triphosphates, adenosine triphosphate and guanosine triphosphate. And the more these adenosine triphosphates and guanosine triphosphates are used already, if they are already used in phosphorylation reactions, then what will be left will be the diphosphates. And so those diphosphates in high amounts give the signal that there is the need for the production of energy for the cell, so that they activate the glutamate dehydrogenase to carry out deamination in the amino acids in order to have the carbon chains in the form of keto acids to produce the carbohydrates and the fatty acids, which will then go into the TCA cycle to generate the energy and even go further maybe into the mitochondrion through other processes, electron transport chain, to produce more energy in the form of ATP and guanosine triphosphate GTP. So the same way, if there are, a lot of ATP and GTP in the cell, then there will be no need to be producing more cellular energy. So they will rather be serving as inhibitors. They will rather slow down the activity of this enzyme, glutamate dehydrogenase. They will rather slow that down. Hormones, they serve as regulators. So there are enzymes like this guanosine, this glutamate dehydrogenase. It can be hormone sensitive. So there are other processes by which some of these hormones regulate the activity of these enzymes. Right. Now, we're looking at oxidative deamination by amino acid oxidases. So what we saw, the oxidative deamination we saw before this was by glutamate dehydrogenase. And you saw that process. If you go back to it, you saw it like from L-glutamate, you have some transition state, alpha aminoglutarate, before you are getting alpha ketoglutarate in that process. That is the process that glutamate dehydrogenase was carrying out. But this time around, we are looking at other enzymes, which are oxidases. So they carry out oxidation reactions like the L-amino acid oxidase, which is much simpler. These ones will act with flavin nucleotides as the monosaccharide, as the coenzymes. So you have the L-amino acid with flavin mononucleotide acting as the coenzyme. If you go back and you look at the glutamate dehydrogenase, what was the coenzyme there? The coenzyme was NADP plus. So you have, let's say, nicotinamide nucleotide. Here you are seeing a flavin nucleotide that is acting as the coenzyme. And that too will go into the formation of a keto acid with the production of ammonia. And of course, because it's an oxidation reaction, the coenzyme, the flavin mononucleotide, gets reduced. So you see, in a reduced form, after it receives hydrides from the amino acid, you are seeing it in its reduced form. But it must continue to act on more molecules. And in that process, you have another enzyme, which is catalase, which is involved in the oxidation of the reduced flavin nucleotide. So in that process, you are having oxygen coming in. Catalase is acting in that process of getting the flavin nucleotide back to its original oxidized form so that it can carry out the reaction further. So you have that cycle. Flavin nucleotide gets reduced whilst you have the L-amino acid getting oxidized, and that reduced flavin nucleotide itself gets oxidized back by the action of another enzyme, catalase, in that process. Now, I think I mentioned to you that in biological systems, you have these DNL isomers of amino acids. And most of the times, it is the L amino acids that you find in larger amounts. And they actually are the active forms of the amino acids. I think I mentioned that to you. And that they act like biological switches. L-amino acids act like the active form on to switch on a certain function, whilst the D-amino acids switch off, see, because it's an inactive form. And I also mentioned to you that in the metabolism of amino acids, you find that when you have D-amino acids, which a lot of times can be gotten from diet, from plants. In the process of metabolism, you have these D-amino acids converted into L-amino acids before they undergo further processes. So you see, this is a D-amino acid, and this D-amino acid, yes, undergo similar process of the formation of a keto acid, which means there is that process of deamination that can occur on the D-amino acid directly. Some hydrolytic reaction is involved in the removal of the amine group with the activity of D-amino acid oxidase. Here too, the coenzyme is flavin adenine dinucleotide. And once it is the D-amino acid that is getting oxidized to give you the alpha keto acid, it means the coenzyme gets reduced. So you now have from FAD, you have FADH2. That's the reduced form of the coenzyme. The alpha keto acid can then undergo a process of transamination to produce L-amino acids. So in that process, you have an L-amino acid that is going to transfer its amine group and forms an alpha keto acid. And this alpha keto acid, which was formed from the D-amino acid, will then receive that amine group to become an L-amino acid. So there is that conversion of a D-amino acid into L-amino acid. Or from the keto acids, we saw already, the keto acids can go into the synthesis of carbohydrates. So you see, you can go into glucose, fat synthesis, and that is going to generate energy. So that is D-amino acids. Before this, if you pay attention to all the slides, you see that all the amino acids that we're referring to were all L-amino acid, L-amino acid, L-amino acid. So D-amino acid, you can have the conversion of it into L-amino acid, and then it can undergo further processes. Now you can have non-oxidative deamination by different processes. These we are looking at non-oxidative deamination by dehydratase. So it's an enzyme, meaning it's going to be involved in what? Dehydration, together with removal of amine group. And this will be with amino acids serine, threonine, homoserine. Take these amino acids and see what is common with them. And you see in the side chain, they have a hydroxyl group, OH, OH, OH. They undergo this specific process of catabolism, non-oxidative deamination. And the enzyme in that process is a dehydratase. So there will be that removal of the amine group, and there will also be some process of dehydration in that process. In the end, it will also produce alpha keto acids. So you see, once you are having amino acids getting deaminated, amine group being removed from them, the product is going to be a keto acid. We can also have non-oxidative deamination by desulfhydrases. And this is particular with cysteine. And what is it with cysteine? Cysteine is one of the amino acids with what? Sulfur in it. We saw two amino acids, methionine and then cysteine. And cysteine is having what? Some sulfur hydral group. So the removal of the sulfur together with the removal of the amino group from cysteine is carried out by the enzyme desulfhydrases, because sulfur 2 is also toxic. It can also be toxic if it's in high amount. So for cysteine, it's not just about the removal of the amine group, but it's also about the removal of the sulfur hydral group as well. So two things are happening at the same time. And take note, what is formed in the end is what? Pyruvate, directly pyruvate. Pyruvate is also a keto acid, but it's a smaller molecule than alpha ketoglutarate. So pyruvate you will find at the beginning of the TCA cycle. Alpha ketoglutarate is somewhere along the cycle. So pyruvate is also what you get from, say, the process of glycolysis that goes into the TCA cycle. So you see, cysteine, which is an amino acid, is producing pyruvate, and pyruvate goes into the TCA cycle and produces energy for you. Then we take histidases. So you can have a histidase. This is acting particularly on which amino acid? Histidine. And histidine is having a special ring in its side chain. Don't confuse this ring to classify histidine as an aromatic amino acid. It is not. We saw 3 aromatic amino acids: phenylalanine, tyrosine, and tryptophan. Their rings are 6-sided rings. Tryptophan has 2 rings fused together. It has a 6-sided ring and a 5-sided ring fused together. But you saw that phenylalanine is having a 6-sided ring, and just the addition of a hydroxyl group on that ring will form tyrosine. Those rings are different from what you see here. This is like some nitrogen-containing ring, some azo ring. And this ring is referred to as an imidazole ring. Now we see just removal of this amine group. The removal of this amine group is deamination. You have what is referred to as urocanate. So the amine group free, and you have the structure urocanate. And histidase is what is catalyzing that process of deamination of histidine. Urocanate, also known as urocanic acid, actually in the liver can get, recycled to glutamic acid. So you have that. Once you have the glutamic acid, it's just like how you were having the other process of the alpha ketoglutarate receiving an amine group and becoming a glutamic acid. Here you have urocanic acid or urocanate that then undergoes transformation in the liver to form glutamic acid. In some disease conditions, like we have some form of a genetic disorder that can result into maybe elevated amounts of this urocanic acid, which will be released in urine, and that will be referred to as urocanic acidosis. So you have some condition of a sort like that. You can go and do some research on it and see. You have some urocanic acidosis where you are having accumulation of urocanic acid due to some genetic disorder. So this urocanic acid will be found in elevated amounts in urine. All right. Now we can take also, let's take the smallest amino acid and look at the different pathways that these amino acid can undergo as far as its catabolism is concerned, even to some extent some anabolism of it. So you begin to appreciate the fact that it's not just some straightforward reaction. One thing occurs with a molecule and that is it. But different things can occur with one amino acid. So we're taking the very smallest of the amino acids. What can happen? Now we take so that's glycine. That is in that rectangle. And take note, some of these arrows are reversible. So far, it is only one arrow over here, which is just in one direction. Okay, there's another one here also. These ones are also in one direction. But pay attention to those ones that are reversible so that you know that those reactions can occur both ways. And what do you see? From glycine, the very first thing you see here is that glycine can break down to give you, first amine group is removed. And there can also be decarboxylation. So removal of this COO minus will give you that CO2. It means that you'll be left with just this CH2. That's carbon skeleton over there. And what do you see? You see that there is a tetrahydrofolate, one of the coenzymes that is involved in that reaction. And you remember the tetrahydrofolate is responsible for the transfer of one carbon residues. So yes, we have this CH2 group that will be left there, and so you can transfer it onto some other molecule. You are also seeing that in this process, you are having a NADH, which is involved in oxidation reduction reactions. And this is actually acting in the opposite direction, where you have carbon dioxide, CO2, like this CO2 with this amine group combining, reacting between them to give you glycine. So you can have some process by which you are having the synthesis of glycine from smaller molecules, say from the carboxylic acid group of a compound and then an amine group. Then you are having some carbon that is transferred, and you'll be having the synthesis of glycine. And that will be catalyzed by glycine synthase. It's an oxidation reduction reaction that can occur. And so you are having this NADH, NAD plus in that reaction. And again, if that reaction should occur, then of course tetrahydrofolate will be responsible for the transfer of the one carbon group in this process to give you that glycine molecule. If you move on to the second arrow, what's happening there? You see, so from one of the products which was formed along the line was glycosylate. And from glycosylate, you can have the synthesis of glycine. But that synthesis of glycine is under the activity of a transaminase. It's a process of transamination, because glycosylate, you can see there's no amine group on it. It looks more like a keto acid, except that there's no carbon at the end. So it's not, so that end it's like an aldehyde group and a carboxylic acid group at the end there. It means there must be transfer of an amine group onto glycosylate to give you glycine. And the enzyme transaminase, what is its coenzyme? Pyridoxal phosphate. Pyridoxal phosphate. Vitamin B6. And you see the red mark there is telling you that in the deficiency of this coenzyme, this vitamin B6, what will happen is that you'll be having accumulation of this glycosylate, because the glycosylate cannot go into the synthesis of glycine since you are having deficiency of this vitamin. In the end, you will have what is referred to as hyperoxaluria. So, because what will happen is that once you are having accumulation of glycosylate, that glycosylate will now have only the path of now producing oxalic acid or oxalate, which will be excreted in urine. That is why you are having what is referred to as hyperoxaluria. And also, another pathway that glycosylate could undertake was for it to undergo some oxidation reaction to form some formic acid, which quickly will go into the synthesis of tetrahydrofolates. But that too can also be hampered, inhibited, and which means that is also going to. So that's going to be like adding up to the accumulation of glycosylate. And you have just this pathway, the production of oxaluria. And so you have a lot of these oxalates or oxalic acid, which will then be detected in urine in high amounts. You can have, for instance, from 309. So 309 can break down under the activity of 309 aldolase to give you glycine. So that's an amino acid from which another amino acid is being produced. Or glycine itself can go into the synthesis of serine. That's by the addition of CH2OH on that alpha carbon. You have serine. In that process, you have an enzyme, serine hydroxymethyltransferase. The coenzyme there is what? Tetrahydrofolates. We saw the activity of tetrahydrofolates to be transferring one carbon groups. CH2OH is a one carbon group. And serine can also undergo further reactions like maybe the loss of this carboxyl group, that's by decarboxylation. And that will result into ethanolamine. Ethanolamine can go into the synthesis of other molecules, like some even neurotransmitters, for instance, and some complexes of lipids. You can have ethanolamine going there. Or serine, of course, after you have a dehydratase, we saw that reaction. Serine dehydratase, it goes into the production of pyruvate, and that pyruvate can go into the synthesis of glucose, or it goes into the tricarboxylic acid cycle to generate energy. The ethanolamine, after deamination, will give you glycocholates, which will go into the synthesis of glycosylate. So you see, it's a web of reactions that can happen with that small amino acid. We are just looking at the smallest, glycine. A web of reactions are occurring. So again, it's not that you have to, chew all of this in your head, please. This is for you to appreciate the fact that one amino acid undergoes different reaction pathways, not only one. So each and every amino acid that you take, it can undergo different reaction pathways. You studied metabolism of proteins, and you ended at proteins break down to amino acids. Now, you are increasing your knowledge to know that that amino acid can undergo different reactions. That's all you are doing. Don't get discouraged and all of that, intimidated, and just be happy you are learning new things. That's all. And don't think that I'll bring something like the huge reaction pathways like these, and I'll be asking, oh, come on. You are not biochemistry major students. You are just doing biochemistry as a course. But you have to, have a broad-based idea about these things. So be happy you are learning it. All right. Now we can take something like phenylalanine metabolism. So phenylalanine tyrosine. So these are now aromatic amino acids. And quickly, all the things which are in green shades, they are enzymes that are catalyzing various reactions. So you see, you have reaction 1, 2, 3, 4. The reactions you are seeing, they are also numbered on the arrows up to reaction 6. So each of these reactions are being catalyzed by different enzymes in green. And you see from tyrosine, from phenylalanine, what do you see? Just by adding a hydroxyl group onto the side chain of phenylalanine, you have tyrosine. And that enzyme is what? Phenylalanine hydroxylase. Because of this, we have what we refer to as sparing action of tyrosine. Sparing action of tyrosine, which is to say that, actually, tyrosine, it is can be synthesized. So if you have phenylalanine, that from it will get hydrolyzed to give you tyrosine. Then there will not be the need to be consuming, tyrosine because of phenylalanine's presence. So you have this, or to say that if you have enough, tyrosine already, synthesized in the body, then of course the consumption of phenylalanine, for instance, is sort of useless because you already have tyrosine there. Then another thing that you should pay attention to is that you see all of these reactions where they end. You see there is a formation of fumarate. Fumarate is one of the intermediates you find in the TCA cycle. So you see, fumarate goes to TCA cycle, can go into the synthesis of glucose. And we also saw already that you have the synthesis of keto acids. Now, when you have keto acids that are binding together, you begin to have what you refer to as acetoacetates. And these ones will go into the synthesis of fats. These are keto acids. So you see, phenylalanine, tyrosine, can go into the synthesis of carbohydrates as well as into the synthesis of keto acids that go into the synthesis of fats. Because of that, these amino acids, together with some other amino acids, are referred to as both glucogenic and ketogenic amino acids. There are some amino acids that will generate only maybe ketone groups that will generate only ketoacetates. Think leucine, lysine, for instance. Those are exclusively ketogenic. But you see, phenylalanine, tyrosine, and even tryptophan, the aromatic amino acids are both glucogenic and ketogenic because they can produce both, carbohydrates and acetoacetates that go into the synthesis of fats. Right. Now, what can happen with, phenylalanine if there is some deficiency, something wrong with the metabolism of phenylalanine? You see, phenylalanine can get hydrolyzed to give you tyrosine. And the enzyme involved there was phenylalanine hydroxylase. The red sign you see on that arrow only is referring to deficiency of phenylalanine hydroxylase. That can result into the condition phenylketonuria. It means there's going to be accumulation of phenylalanine. When you don't have phenylalanine, some of it going into the synthesis of tyrosine. That condition can result into some nervous system breakdown. It results in maybe some mental retardation, hypopigmentation, like albinism. You see some condition of the sort like that. Of course, there are further other reactions that phenylalanine can undergo, transamination to give you phenylpyruvate. So when it loses the amine group, it becomes phenylpyruvate and begins to form some acetates and even some phenylacetyl glutamine in the end. All right, so that will bring us to the end of today's catabolism of amino acids. I'm expecting that there will be a lot of questions. There will be a lot of reactions. The floor is open. You can write in the chat. You can raise your hand, and then I will enable your mic, call your name, and then you speak.